We're still in chapter 24, and we're going to move into metabolism. And metabolism is the sum of all biochemical reactions inside a cell involving nutrients. We have anabolism and catabolism. And anabolism is the synthesis of large molecules from smaller ones. For example, the synthesis of proteins from amino acids. And catabolism is the hydrolysis of complex structures to simpler ones, like the breakdown of proteins into amino acids. We're going to really start talking about cellular respiration in the third video. We're not really going to get into it too much in this video, but cellular respiration is the catabolic breakdown of food fuels whereby energy from food is going to be captured to form ATP in cells. Now, the goal of cellular respiration is to trap chemical energy in ATP. Energy can also be stored in glycogen and fats, which can be broken down later. Then we have this term phosphorylation. And phosphorylation occurs when enzymes shift high energy phosphate groups of ATP to other molecules. Phosphorylated molecules then become activated to perform other cellular functions. There are 3 stages in processing nutrients. We have stage 1, digestion, absorption, and transport to tissues. Stage 2 is cellular processing in cytoplasm. This would be, if it's synthesis, it could be of lipids, proteins, and glycogen, or catabolism, as in the case of glycolysis, where we're going to convert glucose into pyruvic acid and then eventually acetyl-CoA. And then we have stage 3, which is the oxidative breakdown of intermediates into carbon dioxide, water, and ATP. And this is going to occur in the mitochondria. And this is just kind of going over the different stages. I left this up here. I didn't really ask any questions about the specific stages, but just in general, stage 1, trying to find my cursor, there we go. Nutrients are digested. They're digested into absorbable units. I lost my cursor already. There we go. And then they're absorbed into the blood, transported to the tissues. So we have proteins being digested into amino acids, carbohydrates into glucose and other sugars, fats into glycerol and fatty acids. Then in tissue cells, we may either be building or breaking down. If we're building, nutrients may be built into macromolecules. If we're breaking down, they may be partially broken down. And then in glycolysis, that would be catabolism, or the breaking down part. And this is a major catabolic pathway, and it ends with pyruvate. And then finally, we have stage 3, which is in the mitochondria. There we go. And this is the oxidative breakdown of stage 2 products. Carbon dioxide is released, and some ATP is formed. We also end up forming water. Now we're going to talk about oxidation reduction reactions and the role of coenzymes. Oxidation reactions involve the gain of oxygen or the loss of hydrogen ions and their electrons. And then we have this oxidation reduction or redox reactions, and I'm going to refer to them as redox reactions because that's how I learned it. Oxidized substances lose electrons and energy. Reduced substances gain electrons and energy. And this was somewhat counterintuitive to me when I learned this, because usually when I think of reduced, I don't think of gaining anything. So the way I look at this is I look at it as gaining something negative. Electrons have a negative charge. So if you gain a negative, let's say you gain debt, it's going to reduce the amount of money that you have. So I don't know if that helps anybody out, but it helps me. Redox reactions are catalyzed by enzymes that usually require a B vitamin coenzyme. We have dehydrogenases, which will catalyze removal of hydrogen ions, and oxidases, which will catalyze the transfer of oxygen. 2 important coenzymes that act as hydrogen or electron acceptors in the oxidative pathway are Nicotinamide adenine dinucleotide, or NAD. It's NAD plus. I probably would just refer to it as NAD, though I probably should say NAD plus. And flavin adenine dinucleotide, which is FAD. And this is just showing an example of oxidation occurring. So we have succinic acid over here on the left, and succinic acid is this molecule. 4-carbon molecule, it has these 2 hydrogens here. FAD is going to come in here, and it is going to, FAD is going to become reduced, but this is going to become oxidized. And the oxidized version of succinic acid is fumaric acid. And you can see the hydrogens are gone, and now there's a double bond between the 2 carbons. And now instead of having FAD, we have FADH2, the reduced form. There are 2 mechanisms that are used to make ATP from captured energy that is liberated during cellular respiration. The first is substrate level phosphorylation. This one is not, this type of this mechanism does not make nearly as much ATP as the other mechanism does. But with this mechanism, high energy phosphate groups are directly transferred from phosphorylated substrates to ADP. This occurs twice in glycolysis and once in Krebs. And the necessary enzymes are in the cytosol for glycolysis and in the mitochondria for the Krebs cycle. And this is just showing substrate level phosphorylation. I'm trying to get a cursor. There we go. We have a substrate with a high energy phosphate kind of hanging out. And then we have our enzyme here and ADP. This enzyme catalyzes the transfer of that phosphate from here to here, giving us ATP and a product. The other mechanism is oxidative phosphorylation. This is a much more complex process, but it produces most of the ATP. It's a chemiosmotic process. This means that it couples the movement of substances across membranes to chemical reactions. Energy released from the oxidation of food is used to pump protons across the intermitochondrial matrix. This is going to create a steep proton concentration gradient. And we're going to talk about this in more detail later on, don't worry. As protons flow back through ATP synthase membrane channel protein, energy from the flow is used to phosphorylate ADP. So, and like I said, we're going to talk about this actual thing later on in another talk. It'll actually be the next video 3. But protons are going to be pumped across this space, and they're going to end up here in the inner mitochondrial membrane. It's going to create a proton concentration gradient here. And then protons are going to want to flow down their concentration gradient back to the other side. And when they do, they go through this ATP synthase. And this energy ends up phosphorylating ADP to ATP. And I don't want to get into too much detail right now. This is really just an introduction to this topic. We have carbohydrate metabolism. When glucose enters a cell, it's going to be phosphorylated to glucose 6-phosphate. Most cells lack enzymes for the reverse reaction, so glucose becomes trapped inside the cell. Only cells found in the intestine, kidney, and liver can do the reverse reaction and release the glucose. And this is going to keep intracellular glucose concentrations low, which is going to ensure continued glucose entry. What are redox reactions? Is your question. That is all I have for you.